Salmon Escapement Assessment is a core activity of DFO in support of conservation and management of salmon stocks and habitat for sustainable benefits. Salmon escapement surveys provide information about the number of adult salmonids that reach the spawning grounds and the proportion that spawn successfully. Fisheries managers use the term escapement to describe the number of fish that escape the various fisheries. Stock assessment has the primary responsibility for planning and coordinating escapement assessment activities and programs, and for management and reporting of the information that is derived from them. Stock assessment conducts a small percentage of the data collection activities. Delivery of escapement assessment programs requires multitasking of DFO personnel and partnering with external groups. The size of harvests taken by the various fisheries is the major factor affecting the number of fish that return to spawn. Habitat damage also reduces fish numbers. Enhancement or habitat improvement projects can help more fish reach the spawning grounds. Information about stream habitat, such as obstructions, changes in stream beds, pollution, and other potential problems are recorded as part of the survey. All these factors interact to affect the escapement in a single year or over many years. The results of previous surveys can help you recognize trends in relative abundance of various fish populations. For some streams, records go back many years. Counting spawning fish is easier in some streams than others. Some species, like coho, are elusive and very difficult to count. Fisheries biologists sometimes use an easily surveyed stream as an indicator or index stream for a general area, when other streams are too difficult to survey. There are times during the spawning season when stream conditions will not be suitable for a survey. Often there are high flows or floods during the spawning season. Walking the banks then can be dangerous, and fish are hard to see when water clarity is poor. However, high water flows trigger most species to begin migrating to spawning grounds. For many salmon runs, the best time to do the first count is right after the first freshet, as water levels begin to drop and visibility improves. The timing of salmonid migration and spawning depends on the species, the run, and the geographic location of the stream. Adults may migrate to the spawning grounds weeks or months before they begin to spawn. Spawners of various species can be found in streams throughout the year.
Some of the spawner survey objectives are: where are the fish spawning in the river and how many are there? And how does the distribution of spawners in the river compare to previous years? And can we identify the regularly used spawning habitats? Another question commonly asked is the hatchery program reaching its objectives? And how has the spawner abundance varied from recent years? As well as how does the spawner abundance compare to the expectations for the stream's capacity?
An enhancement project can be assessed using a spawner survey. Hatchery staff usually mark a proportion of the fry they release. When fish return to spawn, dead spawners can be recovered, data recorded from the marked ones, and that data used to estimate survival rates for the various groups. The goal of salmon escapement assessment is to collect accurate and consistent field data related to salmon stock assessment activities and environmental conditions. Ultimately, this information could be used to produce credible, defensible, and quantifiable escapement estimates. This is accomplished by using consistent survey and sampling techniques. The basis of these techniques is the stream inspection log. Using this document ensures consistent data and repeatable observations over time. The data to be entered: survey start and end time, environmental conditions, which include percent bankful. This is meant to describe the stream conditions at the time of the survey. Is the water level below or above normal for this time of year? Gauge. This measurement may take the place of percent bankful if a gauge is in place on the stream. Water temperature in degrees Celsius. Temperature is very important to aquatic life. Most aquatic organisms are cold-blooded, so their body temperatures are the same as the water. When the water temperature increases, the concentration of dissolved oxygen decreases. As the water temperature rises, animals use oxygen at a faster rate. Their metabolic rate doubles with each 10 degrees Celsius increase. Plants grow faster and produce more oxygen. However, their decomposition consumes more oxygen. As the water temperature increases, animals become stressed and more likely to succumb to contaminants, parasites, and disease. Organisms die when they cannot adapt to the new conditions. Water clarity. How far into the water can you see? Water turbidity describes the clarity of the water and to what depth fish may be visible. A tape measure is placed vertically into a deep, still pool, being careful not to disturb the sediment on the stream bed. Once the tip of the measuring stick cannot be seen, or it touches the stream bed, the measurement at the surface of the water is recorded. This indicates the depth of visibility of the stream. Be sure that if you can see to the bottom of the stream or river, check the last box, 3 meters or to bottom. Brightness. How bright is it? There may be days when there is 100% cloud cover, but it is still bright. Percent cloudy. How much of the sky is covered by clouds? Precipitation type. Rain, snow, or none. Intensity. How hard is it raining or snowing? Live counts. Live counts are the observations of live fish in the segment that is being observed. They are the count of the species present, not an estimate. Dead recovery. Each carcass encountered should be counted, identified by sex if possible, females assessed for spawning success. Make sure that the columns for percent spawned add up to the total numbers of females recovered. Biological samples. Length, scales, otoliths, opercular punches for DNA are sometimes required for different streams and recorded differently for each species. Chinook and coho samples are to be recorded on a separate field note from the live count field note. Each sampled fish should be included as a carcass recovered. For example, if 7 Chinook females are encountered and only 5 are sampled, 5 rows are filled out on the carcass recovery sampling data field note, and 7 is recorded under females on the total carcass recovery field note. For DNA samples, it should be recorded in the comments section on the sample field notes that they were taken.
The type of spawner survey method to use really depends upon what the objectives are. Generally, we have 6 types of spawner survey methods that vary by the quality of the spawner abundance estimate. Type 1 system develops high quality estimates where individual fish are counted as they migrate past a point, and an example of a method is a fence program.
This method is appropriate for larger streams that have larger fish populations. A man-made structure is stretched across the stream with a single small opening placed in front of a hut on stilts in the stream. Migrating fish are forced to the opening so that the observer in the hut may count them. As fish migrate 24 hours per day, the hut must be manned around the clock.
Type 2 also develops high quality estimates, and that's where we count a proportion of the fish in a river, and examples are mark recapture programs, calibrated multiple survey methods, a Didson method, as well as fish wheel mark recapture methods.
This is the application site, a integral part of the market capture program. Here we apply tags to the fish, and once they are tagged, we release them and they migrate upstream to spawn. After they're done spawning, after the fish die, we do dead pitch on them and get our tag to untag ratio, which is a integral part of the market capture estimate. We only mark the sockeye salmon here because a market capture estimate requires quite a large population to be effective. Other species such as Chinook and Coho also come in this system, but they usually spawn in smaller areas that can easily be counted using a fence or floats or helicopter flights.
This particular system is called the Ditson Sonar Imaging System, and it was developed for the U.S. Navy by the Applied Physics Lab in Seattle. What it does is bridges the gap between sonar and video. So it takes the sonar waves that are coming back to the transducer and puts them through lenses, which is what a camera does. And that then gives you the perception of video, and you can actually see movement of fish. This is called a fixed location system. So in other words, you're near shore and shooting perpendicular out into the flow, and you want to be able to cover the area that fish passage is occurring. If you're dealt with a river that's very wide, you could move fish to where you want to count them, which is done with weirs. And then in conjunction with the weir, you would have your sonar system on the upstream side of the weir, and then counting the fish as they're coming through. What we want is that the fish are actively migrating. And what we don't want to see is fish that are milling, because milling produces obviously more counts than what you would want, because then you would have to know what direction they're traveling. Are they going upstream or are they going downstream? So then when we look for a site, it'll be low in the river system where they're first coming up.
Types 3 to 5, they produce medium quality estimates, and that's where we count an unknown proportion of the fish in a river, and it typically produces an index of the abundance. And some of those examples are stream surveys where there's no calibration information, or fish wheel methods where we just have an estimate of how many fish are caught and how much fishing time was used to calculate the catch per unit effort.
Stream walk. This technique is appropriate for small streams that have deep undercuts. One team consists of a poker and a spotter. The poker walks in the stream, being careful not to disturb any existing reds or fish that are in the stream. Because it is difficult for a poker to see the fish, the spotter keeps watch in the stream channel and keeps track of how many fish are found. Ideally, use two teams, one on each side of the stream, and a recorder, so the poker and spotter may concentrate on finding the fish. Bank walk. Similar to the stream walk, the bank walk uses teams that walk the banks, not in the stream. Observers keep track themselves and meet to compare notes at regular intervals. Aerial count. As this method requires equipment not available to community groups, it is generally done by fisheries and oceans technicians and their contractors. Observers fly over the streams in helicopters and are trained in counting clusters of fish.
Type 6 is a low quality method which just indicates if fish are present. So it's just a record of whether fish were observed, but the method really has low utility other than just saying that there were fish there. It doesn't typically collect enough information to indicate that they weren't there. What type of spawner survey method to use? Well, types 1 and 2 really get at the questions of how does the spawner abundance compare to expectations for the stream's capacity? Or how has the spawner abundance varied from recent years? And other distribution objectives when the stream survey methods are used. So, for example, how many fish went past a certain point, or how many were in this segment of the river. Types 3 to 5 really get at how has the spawner abundance varied at recent years, so we can tell some changes through time. It also provides detailed information about where the fish are spawning in the river, and also how the distribution of spawning fish may have changed from earlier years. And it also helps us to identify the regularly used spawning habitats.
Salmon escapement data constitute an important corporate knowledge which must be maintained and accessible to all Canadians.